Hello everyone, this is Rahul, and today we are going to have discussion on one of the most important topics of molecular genetics, that is homologous recombination. Before beginning our discussion, let me suggest you a few books. For present lesson, I have referred these four books, out of which Genomes 3 by T.A. Brown and Molecular Biology of the Gene by Watson is one of my favorite books of molecular biology. You can also refer Molecular Biology of the Cell by Alberts and others, or Levin's Genes. These two books are equally good. If you are just a beginner, you can refer Karp's Cell and Molecular Biology or Cooper's The Cell, a Molecular Approach. If you are interested in learning advanced molecular biology, you can also refer Molecular Cell Biology by Lourdes. So let us begin our discussion. As Watson says, all DNA is recombinant DNA. Genetic exchange works constantly to blend and rearrange chromosomes, most obviously during meiosis, when homologous chromosomes pair prior to the first nuclear division. During this pairing, genetic exchange between the chromosomes occurs. This exchange, classically termed crossing over, is one of the results of homologous recombination. This recombination involves the physical exchange of DNA sequences between the chromosomes. The frequency of crossing over two genes on the same chromosome depends on the physical distance between these genes, with long distances giving the highest frequencies of exchange. As you can see in this slide, here it is shown different types of recombination. The definition which I gave just right now is the definition of homologous recombination. Homologous recombination may occur between two different DNA molecules or within same DNA molecule. Apart from homologous recombination, there are two other types of recombination too. The first one is site-specific recombination, where a short region of homology is required, and transposition. For transposition, there is no need of homology and it is generally spontaneous and random. So let us see what is homologous recombination in more detail. Elegant early experiments using heavy isotopes of atoms incorporated into DNA provided the first molecular view of the process of homologous recombination. This is the same approach used by Michelson and Stahl to show that DNA replicates in a semi-conservative manner. Key steps of homologous recombination shared by these models include: the first step is alignment of two homologous DNA molecules. By homologous, we mean that DNA sequences are identical or nearly identical for a region of at least 100 base pairs or so. Despite this high degree of similarity, DNA molecules can have small regions of sequence difference and may, for example, carry different sequence variants known as alleles of the same gene. The second step is the introduction of breaks in the DNA. The breaks may occur in one DNA strand or involves both DNA strands. The nature of these breaks is the feature that largely distinguishes the two models described in the subsequent slides. The third step is the formation of initial short regions of base pairing between the two recombining DNA molecules. This pairing occurs when a single-stranded DNA originating from one parental molecule pairs with its complementary strand in the homologous duplex DNA molecule. This step is called strand invasion. As a result of strand invasion, the two DNA molecules become connected by crossing DNA strands. This cross structure is called a holiday junction. Movement of the holiday junction is the fourth step. A holiday junction moves along the DNA by the repeated melting and formation of base pairs. Each time the junction moves, base pairs are broken in the parental DNA molecules, while identical base pairs are formed in the recombination intermediate. This process is called branch migration. The final step is the cleavage of the holiday junction. Cutting the DNA strands within the holiday junction regenerates two separate duplex DNA molecules and therefore finishes genetic exchange. This process is called resolution. As we will see, which of the two pairs of DNA strands in the holiday junction are cut during resolution has a large impact on the extent of DNA exchange that occurs between the two recombining molecules. All the steps I just explained is shown in present diagram step by step. The structure which I called holiday junction, when it is shown in a simplified plane by twisting it in a 2D manner, the structure comes in the form of a chi Roman letter. Hence this structure is also called chi form as shown in this figure. Now there are various schemes for initiation of homologous recombination. The original model says that first nicks are formed at equivalent positions on homologous DNA molecules, then the facing strands get exchanged. Later, Messelson and Rading, two workers, did a little modification in the original model where they suggested that nick is formed first in one DNA molecule only, then a strand after formation of nick invades the facing and homologous DNA molecule, leading to projection of the uncut DNA strand of the facing DNA molecule to touch the homologous DNA molecule where the nick was formed. When this projection, also known as a D loop, enters the parallel DNA molecule, then another nick is formed in this D loop, leading to formation of heteroduplex. This latter model is quite convincing and has been accepted by many workers. One thing you must have noticed that in all previous models, it has been stated that nick is formed in just one strand of a DNA molecule. But during crossing over, you might have read that a portion of DNA or a portion of gene is exchanged, leading to formation of recombinant. So if this has to be occurred, then there must be exchange of both of the strands. So there is one more model, that is homologous recombination by double-strand break repair. Homologous recombination is often initiated by double-stranded breaks in DNA. As shown in this slide, you can see the whole process of double-stranded break repair model for homologous recombination. The common model describing this type of genetic exchange reaction is the double-stranded break repair pathway. As with the holiday model, this pathway starts with aligned homologous chromosomes, but in this case, the initiating event is the introduction of a double-stranded break in one of the two DNA molecules. The other DNA duplex remains intact. After introduction of the double-stranded break, a DNA cleaving enzyme sequentially degrades the broken DNA molecule to generate regions of single-stranded DNA. The process in detail will be discussed in next slide. This processing creates single-stranded extensions known as single-stranded DNA tails. On the broken DNA molecules, these single-stranded DNA tails terminate with 3' end. In some cases, both strands at a double-stranded break are processed, whereas in other cases, only the 5' terminating strand is degraded. The single-stranded DNA tails generated process then invade the unbroken homologous DNA duplex, which is called strand invasion. This in each case, the invading strand base pairs with its complementary strand in the other DNA molecule. Because the invading strands end with 3' termini, serve as primer for new DNA synthesis. Elongation from these DNA ends using the complementary strand in the homologous duplex as a template serves to regenerate the regions of DNA that were destroyed during the processing of the strands at the break site. How the DNA strand is degraded and how 3' end is produced. This will be explained in next slide. There is involvement of different proteins in this process. If the two original DNA duplexes were not identical in sequence near the site of the break, sequence information could be lost during recombination by the double-stranded break repair pathway. So this is the whole process of homologous recombination by double-strand break repair, and there is a kind of repair called mismatch repair. It is a kind of DNA repair, and process double-strand break is observed and has been documented by different workers. So let us move to the protein machinery involved in the process of